Good day, folks! Today on Science Spotlight, we dive into the microscopic marvels of medicine, gene therapy and stem cell research. Genes are the basic units of heredity. They carry the instructions for making proteins. These instructions are found on structures called chromosomes. This process goes like this: DNA to RNA to protein. Proteins are super important because they do most of the work inside our bodies, like building structures, sending signals, and keeping things running. Ways to do gene therapy, and that is ex vivo and in vivo. Can we edit out disease? Can we grow new organs like spare car parts? Let's find out.
Welcome to today's exploration of gene therapy's unsung heroes, viruses, and vectors. So, let's dive into how these tiny tools help rewrite our genetic code. Viruses are like nature's delivery experts. In gene therapy, they are repurposed as vectors. Vectors are carriers that transport therapeutic genes into human cells. But not all viruses are created equal. Retroviruses, like the human immunodeficiency virus, or in short, HIV, inserts genes permanently into our DNA. But this method can disrupt other genes, posing risk for cancer. Adenoviruses, behind the common cold, deliver genes temporarily but trigger strong immune reactions. Third, adeno-associated viruses are safer, targeting chromosome 19 and used in hemophilia treatments. Finally, herpes viruses target neurons, which are useful for brain disorders. Not a fan of viruses, non-viral vectors offers alternatives. Scientists use lipid bubbles called liposomes, or chemically attached DNA to molecules that cells absorb. Some even try adding a 47th chromosome, but these methods are trickier, less efficient, and harder to deliver. While viral vectors are powerful but risky, non-viral methods are safer but need refinement. Together, they are paving the way for groundbreaking cures. That's it for my part. Thank you for watching and stay curious.
Good day and welcome to Science Spotlight. Today, we're taking a closer look at an amazing discovery: stem cells. These special cells can divide and turn into different types of tissues, giving doctors new hope to treat diseases and injuries in ways never seen before. Well-known figures like Nancy Reagan and Michael J. Fox spoke out about the importance of stem cells research. They believed it could help millions of people who suffer from conditions medicine couldn't cure before. The history of stem cell research started back in 1998 when scientists first isolated stem cells from human embryos. Even though funding was restricted in 2001, research didn't stop. In 2009, President Obama lifted those restrictions, giving new life to the field. From 2012 to 2014, researchers made a big moves, using stem cells to help people with blindness and even figuring out how to rewind cells to an earlier state, opening up even bigger possibilities for healing. Today, stem cells are seen as one of the most powerful tools in medicine, with the potential to repair, rebuild, and restore health like never before. And that's today's quick look at the future of the medical science.
We're now diving into the fascinating world of stem cells, the body's very own lung cells. Stem cells are unique because they are unspecialized, meaning they haven't chosen a role yet. But here's the twist. They can divide and renew for long periods and even transform into specific cell types like muscle, nerve, or blood cells. Three are the major types. First is the totipotent stem cells, which is the most powerful. They can become any cell in the body and the placenta. These appear in the first 1 to 3 days after fertilization. Next is the pluripotent stem cells. From blastocysts can form over 200 cell types but not the placenta. Lastly, multipotent stem cells, found in places like bone marrow or cord blood, are more limited, forming related tissues only. And these two main sources are embryonic stem cells and adult stem cells. Embryonic stem cells are from early stage embryos, while adult stem cells found in various body tissues, even in your skin and brain.